All right, thank you all for attending. So today I want to talk to you about cell spaghetti. So what is it and why does it need untangling? Your body, each one of your bodies is made up of trillions of cells. And inside each one of these tiny cells are millions to billions of proteins that exist in a very crowded environment. And if you could look inside, it might look something like this image in the middle. And if you think of your cell like a factory, the proteins make up all of the different components of this cell or of this factory. So they include all of the workers, all of the machines in the factory, and even the foundation of the factory itself. For example, in our body, we might have some proteins that might act like a pair of scissors and chop up unwanted material that our cell no longer needs. Some proteins might form large networks that span the entire length of our cell and give our cell a framework and some structural rigidity, much like the framework of our houses. And other proteins might actually function like little factories in and of themselves. For example, we have these light blue proteins that are acting like a conveyor belt that are bringing materials to this green protein that is producing new material that our cell requires. And so ideally, all of these different components in our cell would work together in harmony. And this is what allows our cells to live and for life to exist. But sometimes there's problems that occur. In this instance, you have these three different proteins that are all working together, but sometimes they might bump into each other and they might accidentally stick to one another. And then when this happens, they stop functioning and it can lead to problems. And if they stay stuck for too long, what happens is that their structures can actually unravel and they can become entangled with one another and they can form something that looks like a plate of tangled spaghetti noodles. And so this is called protein aggregation. And if this aggregation process remains unchecked inside our cells, it can lead to cell death or to the development of neurological disorders. But we live pretty long lives, and I'll tell you, this occurs in all of our cells all of the times. So somehow our cells have figured out a way to reverse this process. So I'd like to introduce you to the hero of the presentation, which is a special type of protein called a disaggregation protein or a disaggregation machine. And the goal of these machines, such as this one in the center, is to find these balls of tangled noodles, bind to them, and pull them apart or separate them out from one another. And this allows these proteins, these misfolded proteins, to then refold into their native structure and carry out their function. But even our heroes need help sometimes. It turns out, as we age, sometimes these machines stop working properly. So whether that's because the machine itself becomes less efficient, or if they simply become overwhelmed with these spaghetti noodles. We're not quite sure. But to complicate matters, we don't even know how these machines work. How do they untangle this mess? We don't even know. So the first step, of course, is, if we want to answer these questions, is to figure out how they work. And that is where I come in. So it is the goal of my research to figure out how one of these machines work. And so today, for the rest of my presentation, I'm going to talk to you about one of these machines, ClpB. So one of the defining features of ClpB is that it has a large central pore that spans the entire length of its structure. So that if you could take the protein and look through it, you would see a hole all the way through. And many people think it uses this distinct feature to untangle these spaghetti noodles. Some people like to envision it that ClpB binds to one of these misfolded proteins, these tangled spaghetti noodles, picks one of them out, grabs onto it very tightly, and pulls it out, kind of like slurping one spaghetti noodle off of a plate. But other people think this cannot happen because it takes too much energy. If you were to consider if you had a plate of tangled spaghetti noodles and you had to pick one out, sometimes it requires a lot of force to pull it out. And so some people think this little blue protein can't do that. It can't exert enough force. So instead it must do something else. Exactly what, they're not sure. But maybe take this plate of spaghetti noodles and slowly spread it out on the plate, loosening these aggregates, which eventually allow the noodles to fall apart. And in principle, this should be pretty easy to figure out. If we could simply just look at the protein, we could do a couple simple measurements. For example, if we could see how far these spaghetti noodles can go into ClpB, do they only go in a little bit, or can these misfolded proteins go all the way through? We could also time these events to tell us how quickly ClpB is functioning, and we could use this to determine if these proteins, if they lose efficiency over time. But there's a problem. The proteins are far too small to see. Even with the best microscopes, they're invisible. So we need to be a little bit clever in how we do these measurements. So in order to do studies like this, we need to use some sort of sensor. And so ideally, we want this sensor to have a couple different properties. We need it to be able to measure short distances. These are distances on the nanometer scale, so very tiny. We need these sensors to respond in real time so that we can see changes in distance as they occur. And of course, we need them easy to see. So it turns out such a sensor exists, and it exists as what are called a fret pair. And these are two specially chosen fluorescent dyes that exhibit some special properties. We'll call it sensor 1 emits green light, so that means it's green, and sensor 2 emits red light. And so it turns out when these two sensors are far away from one another, we only see light from the green one, indicated by the large green star. But then as these two sensors come closer to one another, the green one gives its energy to the red one, therefore we'll see less green light and more red light. So it's actually pretty simple. We can use these sensors, and if we see green light, we know they're far away, and if we see red light, we know they're close. So we can use this change in light as a little spectroscopic ruler to monitor changes over time. And the idea is as follows. I will take ClpB, the disaggregation machine I want to study, and I will put one of the sensors on it. I'll then put the other sensor on the misfolded protein. And so if we only see green light, that means that they're very far away from one another. But as ClpB begins to disaggregate or interact with the spaghetti noodle, we'll see changes in the intensity of light. And by how much red light we see, we can determine how close they can interact. And so in practice, what this might look like is a plot or a trajectory as shown along the top. At time 0, the protein, the ClpB, and the substrate are very far away from one another. So we see no red light whatsoever, and we only see green light. They're more than 10 nanometers away in this part. Then over time, they will diffuse around, and once in a while, ClpB will find a spaghetti noodle. And when it does, we'll see a little flash of red light. And by looking at the characteristics of these flashes of red light, so how intense they are and how long they last, they tell us information about how ClpB is interacting with the noodle, whether the noodle only goes in a little bit or if it goes all the way through the protein. So by looking at many such trajectories of these and applying statistical modeling methods, I will be able to build a picture of how ClpB functions on the molecular level. And together, by using this information, I hope to answer the question, how does ClpB untangle cell spaghetti, in hopes of using this information to better understand this protein aggregation problem and how we can potentially reverse it in the future. Thank you all, and I'm happy to answer any questions.
Thank you. Great talk, Remi. Thanks. San, Weizman Institute. Do you think you can apply this in the future for like amyloid beta plaque disease, like for Alzheimer's diseases, against protein aggregations in like the amyloid beta plaques? Sure.
So your question is, can I use these kind of things to study like the amyloid fibrils that form? We can. I mean, it should be possible. Right now, because I'm just starting this research, I'm trying to keep things as simple as possible. But later on, once I have a better grasp of how to handle all of my data and extract all the meaningful information. That's actually something that we plan to do, is study these more complex aggregates. And these proteins themselves have been used by other researchers to try and address these neurodegenerative disorders. But there's some strange things that occur. Since these proteins, they disaggregate like your amyloid fibers. So you'd think you add more of these proteins, you would get less Alzheimer's. It turns out if you only add a little bit more of these, the Alzheimer's actually progresses more quickly. So this is why I think it's very important to understand how they're actually functioning on a molecular level. Because I mean, we make some very simple assumptions that more disaggregation machines, the better, but it's not true. So I mean, it's very confounding. Thanks.
We don't hear you through it, but the live stream does.
Hello, live stream. Thank you for a very interesting talk. I have a question. What happens after you untangle the spaghetti? I mean, do those protein chains fold together back to a structure, or they just decompose and the cell builds more proteins?
Yeah, thank you for the question. So you're asking what happens to the spaghetti noodle maybe after it comes out the other side? So after this happens, the protein will try and refold. So sometimes the protein will refold into its native structure or its correct structure, but sometimes again, it will misfold or it might find another aggregate. So this actually occurs in a repetitive process. So if the protein doesn't fold correctly, again, the disaggregation machine can find it and again pull it through and try and fix it.
I have one follow-up question. For different types of proteins, you have those different types of machines that unfold the spaghetti entanglements? Or it's one general type of machine that does it?
Okay. So in general, every organism has its own different types of disaggregation machines. And so different cells might use different machines, but there's general types of machines that are present in many different types of life and many different types of cells.
Hey, Slava from Weizman. I know that cells, when they are trying to push spaghetti around, they're using different pore proteins, which are untangling proteins, and then they are putting them back in a form. Is there some kind of a resemblance between these things and transporters, like from ER, from cytosol to endoplasmatic reticulum and so, which are putting the protein, putting the spaghetti, untangling it, and taking the whole thing through?
Yeah, so that's a good question. The question is, there's these these disaggregation machines pull the spaghetti noodles, but there's other proteins that will pull these noodles, but for different purposes, and are they related? So the proteins, well, of course, they'll be different, but there are many domains within these proteins, or regions of them, that will be quite similar. So for example, this protein, inside the pore, it has little hands that can grab onto the noodle and pull it. And so there's many other types of proteins that share very similar features. So of course, in this case, because it's doing substrate translocation, it's very likely that these pore proteins will share some similar features. I'd have to look into the specific proteins to see which mechanisms it uses. But in general, if we can understand this protein, it would help us understand many other proteins as well.
Monica from Weizman, beautiful talk. You just said that if you make more of this aggregation machines, they could accelerate Alzheimer. But what about making those that you have more efficient? A, could this be done? And B, could you read it out with your methodology?
So the good question. So your question, if I understand, is if we make changes to this protein, can this methodology can we see it with this methodology, such as we make the machine more efficient or we use more or less of them? And the answer is yes. So there are many different mutations or changes you can make to the protein to either make it more or less effective. It can be very fast, and how much energy it consumes during these interactions, you can change that. And of course, with this current methodology I'm using, it'll be very easy to see the different changes. So exactly, this is why I'm very interested in this, because it gives us a very clear picture of how the protein changes.
Thank you for the talk, and perhaps I missed it during the talk, but so you described that you can precisely determine when the protein and the noodles are close and when they are apart, and is it enough to know if they are just really doing something, or is it enough to determine the mechanism of?
So what I'll be measuring? Your question is it enough to know if the noodles are close or far apart from one another? So in my experiments, what I'm measuring is the distance between a specific location on ClpB, the disaggregation machine, and a position on the spaghetti noodle. So this way we get to see how close these interactions occur, and we can do some mapping and convert, say, this like three-dimensional space into fret or energy transfer landscape, and we can actually see in three-dimensional space how these noodles are moving. So it does take a bit of work to do that, but that's kind of the idea. By looking at just this one-dimensional data, seeing more or less red light, we can actually get information in three-dimensional space.
How does it know it's aggregated?
So the question is, how does this protein know it's aggregated? That's a great question. So I didn't go into more detail. This is just actually one of the disaggregation machines in a whole family or network of machines that work in harmony. So this machine itself actually recognizes a much smaller machine whose sole job is to identify these aggregates and bind to them. And then this blue one comes along, recognizes the smaller ones, and then it knows where it has to function. But typically, in general, it can recognize these misfolded proteins due to changes in hydrophobicity or how sticky the proteins are. These misfolded proteins tend to be more sticky, and folded proteins are less sticky. Thank you.
Thank you very much.
Thank you.